Good morning, students. Students, am I audible?
Yes, sir.
Right. Today we are going to discuss about virus-free plants. Yesterday we continued, right? We started yesterday. Types of plant tissue culture and applications of plant tissue culture. This topic have been discussed previously, right? Let us continue. Virus-free plants. Actually, virus-free plants are nothing but the plants are produced without a viral infection or the pathogen of virus, right? That is called virus-free plants. So we have discussed about a number of techniques in plant tissue culture, right? So one of the techniques that we are going to apply for making or getting or developing or producing virus-free plant, right? That is called the apical meristem culture. First of all, you should understand what is virus-free plant. It means the plants are not affected by viral pathogen, right? So that is called the virus-free plants. So generally, we are going to apply one of the techniques of plant tissue culture, that is apical meristem culture. What is apical meristem culture? Apical meristem culture is a kind of plant tissue culture by which we can develop free virus-free plant. That means free plants are produced without infection of the pathogen, right? That is called a virus-free plant. That means the plant, it cannot be affected by virus particle or virus, right? It is called a virus-free plant. Now, it is possible to produce a virus-free plant by using one of the techniques of plant tissue culture, that is the apical meristem culture, or it is also known as a shoot apical meristem culture, right? Because the apical meristem is a growing portion of the stem of both root and the shoot, right? Apical meristem, it is a growing point of the shoot system. Now, let me represent a diagrammatic representation of plant. Right now, this is a plant. If you take a plant, the plant consists of, now you know very well, leaf is there and the branches you find, right? And at the tip you will see a growing portion, right? So that is called the apical meristem. I'm talking about, this is a portion where you see, right? This portion is called the apical meristem. Apical meristem is nothing but, it is a growing portion of a stem that is called the apical meristem. Now it is free from the virus particle. So in the point of apical meristem or a growing point of the shoot in which there is no presence of virus particle, right? So here this is the apical portion or the growing point or growing portion of the shoot system is taken out and which is used as an explant for making or developing entire plant. So that is called the virus-free plant. Now just I would like to draw the structure of apical meristem, right? Now when we cut the apical meristem as an explant, right? Now I am going to cut it and used for as explant. Now this is a apical portion. That is a growing point, right? Now I'm going to use it as an explant. Already as we have discussed, explant, it's a part of a plant which is used for plant tissue culture technique in order to develop organ or entire plant in a synthetic medium under controlled condition, right? That is called plant tissue culture. Now, in plant tissue culture, I have taken a small piece of plant that is called X plant. So, X plant, as I said repeatedly, it's a part of a plant which is used for making entire plant or any other organ of plant that is called X plant. Now, I am going to develop entire plant from the X plant by using a synthetic medium or growth medium, right? So, as a result, this X plant is converted into like unorganized mass of tissue, right? This is called tissue. This is a tissue which is having capacity to make shoot system and root system. This callus, as I said, callus is an unorganized mass of tissue. It is able to make or develop to produce both shoot system, shoot, and on the other hand, we can say both root. So this root system and the shoot system can be developed from the unorganized mass of tissue that is called callus. So finally, this callus tissue, which is going to differentiate it into shoot and the root system, right? With the help of a culture medium. So finally, we get a small plant. I mean, a plantlet that is a resin plant, right? So this is a plantlet, and these plantlets are exposed, right, to the environmental condition. Plantlet. Plantlets is nothing but it's a small nursery plant which is growing inside of the test tube, which is grown in a synthetic medium under controlled condition, right? So that is called simply plant tissue culture technique. So one of the plant tissue culture technique which is applicable for making or developing or producing virus-free plant, that is a apical meristem culture. Students, please understand. I'm talking about apical meristem culture. Simply, the apical meristem is a growing point of the stem which is taken as X plant, right? Which is taken as X plant. First one is a growing point of the plant, that is the apical meristem. It is taken as a piece or part of plant that is used for plant tissue culture technique, that is the X plant. So X plant is the second plant. These X plants are utilized for plant tissue culture technique. I mean, it depends some growth substances and culture, media culture, vessel, etc. Right? So after that, after some time, what will happen is this X plant is converted into unorganized mass of tissue that is called callus. So after formation of callus, which is transferred into another culture medium, as a result, we can get shoot and the root system, right? Simply this callus which is going to differentiate on one side shoot system and another side root system. As a result, we obtained it, we got it like a plantlet, a small plantlet. So now these plantlets are grown inside of the test tube. Just imagine now this is a test tube. So the final step of plant tissue culture, these plantlets are exposed to environmental condition, right? It is to be exposed to the environmental condition in a stepwise manner that is called a hardening process. So this is a way by which we can develop, we can produce, we can make virus-free plant. Why I'm saying, because nowadays when you go for cultivation, right? So the crop yielding almost it's going to be affected by any other pathogen, right? There are so many pathogens of the pathogen. You should understand, dear student, please understand. I'm talking about pathogen. Pathogens are nothing but, it may be microorganism, it may be bacteria or fungi or mycoplasma or any other protozoa or prokaryotic cell. which are going to cause disease. They are called pathogen. I repeat once again, pathogen is nothing but an organism. It is having ability to cause disease to other organism, other living organism. That is called the pathogen, right? So I'd like to give some pathogen. The first one is a fungi. The first one is a fungi. Now we have heard about what is fungi, and what are the types of fungi, and what is its general characteristic features, and what are the diseases caused by fungi, right? We have discussed a number of diseases. And one more pathogen, that is bacteria. There are so many diseases or there in plants which are caused by bacteria. Xanthomonas citrate that causes citrus canker disease, right? Cercospora persanata, it's a fungal disease. Just I like to touch every disease, right? So Cercospora persanata, Cercospora persanata, Cercospora persanata. It is one of the fungi that is going to cause Pica disease. Pica disease. Just imagine Pica disease. It is affected in groundnut. So simply, this groundnut plant is affected by one of the fungi that is a Cercospora persanata that causes a kind of disease that is a Pica disease. Then what is its symptoms? There is a black or brownish dots are appeared on the surface of leaf and stem portion, right? So that is called a Pica disease. Why I'm saying here? Because these fungi is a pathogen. The name of the fungi is Cercospora persanata. It is one of the fungi that causes a disease, Pica disease in groundnut plant. So this groundnut yielding is totally collapsed by the entry of Cercospora persanata. So this Cercospora persanata is considered to be one of the pathogen. And next one, bacteria. The bacteria is also known as a pathogen because it is able to cause a disease to some crops, right? In citrus canker, lemon fruit, it is totally affected by Xanthomonas citrate. It is also one of the pathogen, right? That causes a canker disease. So the surface of lemon fruit is totally affected by black dots, right? So commercially it is to be affected by citrus canker disease. So like that, one of the fungi, one of the pathogen, that is a mycoplasma. So mycoplasma and virus. Finally, let us take virus. So these are all the pathogen, but except virus, all these three pathogen can be controlled by using so many chemicals are there, right? So fungal disease can be controlled, and bacterial disease can be controlled, and mycoplasma disease also can be controlled, but viral disease cannot be controlled, right? By using any other fungicide like some chemical agent or involved in controlling and the killing of these microorganisms or pathogen, except to virus. So due to entry of these pathogen in the field of crop, what will happen is totally they are going to collapse. They are going to reduce the yielding capacity, right? And hence the cultivators, they may get a large amount of losses. So in order to prevent such a losses in the field of agriculture, or in order to increase productivity, and in order to supply productivity, or in order to increase yielding capacity, or in order to increase resistant power, we have to depend, we have to prepare, we have to make virus-free plant, right? So all the cultivators who are depending on virus-free plants in order to produce more crops in the field of agriculture. That's why the scientists who concentrated on apical meristem culture, particularly you can say shoot-tip apical meristem culture. Particularly I can say shoot-tip apical meristem culture. Students, please understand, shoot tip. Shoot tip means a stem. It is also known as a stem. The tip of a stem that possesses apical meristem. The tip consists of, or tip is a shoot tip or a stem tip. Both are similar meaning. The shoot tip is nothing but it is a apical meristem. Now this apical meristem is artificially taken into plant tissue culture technique and which is applied for developing virus-free plant. That's why this is a shoot apical meristem culture is a method of plant tissue culture that is going to prevent the losses which are occurring in the field of agriculture. I mean, the pathogenicity can be totally eradicated by using of apical meristem culture, right? So please understand, dear student, virus-free plants can be produced by using apical meristem culture. Simply I can say this is the apical meristem culture which prevents the pathogenicity of virus. Please understand apical meristem culture that prevents the entry of virus particle inside of the plant. Right? So this is simply something about of virus-free plant. Okay. Next we pass on to conservation of plant genetic resource. Before going to discuss about, just I'd like to ask some questions. Yes, students, did you understand? Students? What do you understand about virus-free plant? And which of the following techniques of plant tissue culture is involved in making virus-free plant?
Shoot meristem tip cultures.
Very good. Shoot meristem tip culture. Do you understand the diagram is drawn on the screen?
Yes, sir.
Right? Could you mention the growing point of plant? What is it called? Yes, students. Actually, growing point is also known as the apical portion. Apical portion is also called as a growing point or growing tip of both shoot or stem and root. Dear student, what is another term of growing point of stem and root? Apical portion. Because apex, apical cells are possessed. They are generally free from the pathogenicity. Understand? Right. Next we pass on to conservation of plant genetic resource. That is very interesting topic. Conservation of plant genetic resource. So generally, this conservation of plant genetic resource is divided into two types. Before that, you should understand what is conservation. Conservation is nothing but preservation. Conservation is nothing but preservation. Yes, students, am I audible? Here, answer is hidden in the title, conservation of plant genetic resource. Actually, the plant genetic resource are preserved for future purpose, or for breeding purpose, or for improving crops in future, or in order to do research work, like pollen, seeds, or tissue, or any other organ, or organelle, right? So they are called simply, the biological sources are protected, or preserved, or conserved for long duration, for the purpose of research, for the purpose of breeding, for the purpose of maintaining fertility and food security, or in order to emerge biodiversity, in order to maintain, protect biological diversity, to increase food security, right? So in order to simply conserve or protect the plant materials, are protected or conserved or preserved for future purpose, right? It is called the conservation of plant genetic resource. Dear student, please understand, conservation of plant genetic resource are nothing but the biological materials or the biological sources, like protoplast or cell or DNA or gene or pollen or the tissue or any other thing, right? First of all, let me introduce, right? First of all, protoplast and cell or organelle, right? Protoplast, cell or organelle or any other extracellular matrix, right? And pollen, and one more thing, right? Seed or any other biological materials which are protected or conserved for future purpose, that is called a conservation of plant genetic resource. Now, this conservation or preservation of plant genetic resource are divided into two types. They are, simply I can say, germplasm conservation and the cryo preservation or cryo conservation. Dear student, please understand, the conservation or preservation of plant genetic resources are divided into two types. What are they? The first one is germplasm conservation. Germplasm conservation. Germplasm conservation or preservation. And the second category, cryo preservation. Dear student, please understand, second category is a cryo preservation. Cryo preservation. What is germplasm conservation and what is the cryo preservation or cryo conservation? Both are similar meaning. Preservation and conservation. Storage or protection or savings. So all are similar meaning, right? So first of all, let me take germplasm conservation. Actually, germplasm conservation is a type of conservation of plant genetic resource, right? In which we have to protect pollen grains. So the first thing is pollen. Pollen grains are protected or conserved. Not only that, and the seeds also protected, either pollen grains or seeds. They are protected for future purpose. in order to maintain, viability, and fertility, for improving of hybridization and the craft improvement. For the purpose is nothing but simply in order to improve hybridization technique. And one more thing, hybridization and craft improvement. So in order to increase hybrid vigor in plants and in order to increase craft productivity, the pollen grains and the seeds are conserved. Those plants are producing good or viable seeds. This viability and fertility can be retained by the storage of pollen grain and seeds in plant, right? So that is very important and a very interesting area in germplasm conservation. Simply talking about the germplasm conservation, it is a method of conservation of plant genetic resource that includes pollen grain and seeds. If the pollen grains are viable, so this viability can be retained even if it is stored for long duration, right? For the study of pollen grain, it can be stored. In order to supply the good seeds or very vigorous seeds for next generation, such as seeds can be preserved or conserved with the maintenance of viability and fertility for long duration. Not only that, in order to maintain or in order to increase hybridization and in order to increase craft improvement in plant. That's why these pollen grains and the seeds are protected or stored. Students, do you understand what is germplasm conservation? What do you understand about germplasm conservation? My dear students, here what are going to store? One of you please answer. Pollen grains or pollen
Grains and seeds.
and seeds. Would you like to see the storage of pollen and seeds? My dear students,
Yes, sir.
I have some collections. Now this is a pollen bank. Actually, pollen bank is nothing but it is a bank that consists of simply. Dear students, please understand the storage of pollen in a controlled condition. Artificially, the pollen grains are stored or conserved or preserved for future purpose. I mean to do research work or to study of the general characteristics of pollen and in order to maintain the fertility and viability of the pollen and the seed also, right? So this is one of the pollen bank. The pollen bank is nothing but it is a bank in which you can see the collection of pollen grains of various plants which are available very rarely, right? So that is called a pollen bank. Now one more I am going to show you, that is a seed bank. My dear students, do you like seed bank? Do you like to see seed banks?
Yes,
Now,
sir.
if you go to cultivation, you see there are number of crops are grown in the field of agriculture, right? Here in some places you can see harvest is very high or crop yielding. In some region, if you go to Tanjore, you can see paddy crops are cultivated in large quantity because the soil is very suitable, and so the seed, I mean the pollen grains or the seeds which are cultivated in Tanjore, you can see, right? So let us take only one example, that is the paddy. So if the paddy is, there are a number of paddies on the right, number of varieties in paddy, IR8, J30, so many categories are there, right? So the good seeds or the vigorous seeds are stored for future purpose in order to supply or transfer such a hybrid vigor from one generation to another generation or from one place to another place. So the seeds are also protected or conserved for the future purpose, right? So that is also known as a seed bank. The seed banks are nothing but it is a collection of seeds. Actually, the seeds are stored not for selling, to simply use for future purpose. So that is called a conservation, right? I have given two bank. One is a seed bank and another one is a pollen bank. Just imagine seed bank and the pollen bank. Both of them are nothing but collection of pollen and the seeds for future purpose. So they are the example of germplasm conservation. Please understand, I have given two bank. One is a pollen bank and another one is a seed bank. So in first case, you see there is a small tube that contains a large amount of pollen along with the viability and fertility, right? Why it is stored? In order to increase hybridization and in order to improve crops in the field of agriculture. And why we have collected various seeds and stored and preserved? Because the nature of seeds and the study of seeds and the categories of seeds and the general characteristics of seeds. So all the quality of the seed is also indicated, right? So the quality and the characters of seeds are collected and labeled just inside of the bottle, right? They are which contain seeds. So this is called simply germplasm conservation. In addition, not only the storage of pollen grains and the seed, we can also store, we can also preserve DNA and the gene, right? DNA and the gene. So simply, I'm going to talk about gene bank. What is a gene bank? Actually, gene banks are nothing but it is a collection of gene which are also preserved for the future purpose, to research work or the purpose of research, to study about its character. Just I'm going to show you some example for you. So please understand. Right? Gene bank is nothing but a collection of various genes, because every gene controls a character. That is called one gene, one enzyme concept, right? According to one gene, one enzyme concept, every character is determined by a particular gene. That's why the genes are collected and preserved for future purpose. That is called gene bank. Now I'm going to show you some gene bank. Now you can see there are different plants, different kinds of plants, right? Yes, students, please see. Here you are seeing some plants, and these plants are indicated along with the gene. So the character or a particular character is special. Just imagine, if we take the first plant, right? If we take the first plant or the second plant or whatever it may be, and let us take the middle one, right? So these plants are taken, right? The plant is determined by a particular gene, and its character, the special character of the taken plant, right? Particular character is taken, and this character is determined by a particular gene. Just imagine, right? Now this gene is indicator in the test tube. The test tube contains simply a plant that expresses a very special character, right, to the agriculture field or to the research world, right? So this character is determined by a particular gene. So the gene, the name of the gene is also indicated and labeled inside of the test tube. So that is called a gene bank. So simply a collection of genes from various countries, and they are stored, they are preserved along with the plant characters, right? So that is called a gene bank. And one more I'm going to show you, that is a DNA bank. No, DNA is a genetic material. DNA is a genetic material. DNA bank. No, DNA bank is nothing but it is a collection of DNA of various plants. It is also protected. It is also preserved for long duration. I'm going to show you next one. Because the DNA is a genetic material. Genetic materials are nothing but the total character of a plant is determined by DNA, right? If you take just a groundnut, the groundnut which is able to cause, it is able to produce crops just under the soil. This character is determined by DNA, right? Because DNA is a genetic material. Now, such a genetic material which controls a particular character or expression of both morphological and anatomical character, this character are determined by DNA. Now, DNA is a genetic material. This genetic material is a stable or a constant. It's going to express generation after generation. Now, such a character is also protected in the form of DNA, right? A DNA can be stored like storage of gene, storage of pollen, and storage of seed in a preserved place, right? In a preservation method. So that is called a DNA bank. DNA bank is nothing but it's a collection of DNA, right? So why I'm saying, what is the use of this collection of or this gene bank and the DNA bank? Actually, this gene bank and the DNA bank are essential to maintain, in order to maintain biological diversity. Students, please understand, in order to maintain biological diversity. In addition to that, in order to maintain food security, So this is a role of a gene bank and the DNA bank, right? Student, please imagine. So we have discussed it. There are many banks: seed bank, pollen bank, gene bank, and DNA bank. So this gene bank and DNA bank are involved in order to maintain biological diversity and food security, right? So this is a role of gene bank and DNA bank. Dear students, what do you understand about gene bank and DNA bank? First of all, what do you understand about gene bank? My dear students, could you share about gene bank and DNA bank? What is the use of gene bank? Students, am I audible?
Maintain biological diversity and
food security. And one more thing, what is it? Food security. Food security. Now to maintain food, because nowadays our population is so high, right? We are a second place in world population, right? So in order to supply food material throughout our country or throughout the world, there will be food demand. So in order to maintain food security, we have to conserve, we have to preserve, we have to protect both gene bank and DNA bank. The gene of a good quality, good variety of crops, cereal, or good variety of whether pulses or nuts, or oil yielding plant, or cereal, which are going to be protected. When we protect, what will happen? We can maintain biological diversity and food security. So we cannot face a scarcity of food, right, in future, right? So in order to avoid the demand for food for throughout the world, right, for children or for all the people, right? So in order to simply prevent the food scarcity. So that's why the DNA and the GNA are stored. I mean, the gene and DNA of good variety. Okay, next we pass on to cryo preservation. So far we have discussed only germplasm conservation, my dear students, right? Now I am going to talk about cryo preservation. Actually, cryo preservation is nothing but already I have discussed with you. It is a preservation of protoplast cells or cell organelle, tissues or extracellular matrix or extra. At extreme temperature, at very cooling temperature, minus 196 degrees Celsius, these plant materials are preserved or conserved, right? Called a cryo preservation. So, dear student, please understand, cryo preservation, it is nothing but, it is the preservation of cells, tissues, or organelle or organ at extreme temperature, very cooling temperature. That is called a cryo preservation. Cryo preservation. Simply I can say cryo preservation is also known as cryo conservation. It is also called as cryo conservation. Cryo preservation, it is also called as cryo conservation. What is the temperature that we are using here? Simply minus 196 degrees Celsius. What are protected? It is a preservation of, simply, it is a preservation of protoplast, or cell, or protoplast cells and cell organelles or extracellular matrix or enzyme or any other extracellular matrix. Extracellular matrix. So they are called simply cryo preservation. Cryo preservation, it is a storage of or preservation of following things, such as protoplast, or preservation of cell or cell organelle or enzyme or extracellular matrix, right? Or proteins or in etc. So this is called a preservation or cryo preservation or cryo conservation. Cryo preservation, it is also known as a cryo conservation. This in cryo preservation, there is a temperature minus 196. This is very extreme cooling temperature. In that condition, protoplast cells, cell organelles are protected or conserved for the purpose of future studies, right? So that is called a cryo preservation. Here, cryo preservation depends on the chemical agent, that is nitrogen. So nitrogen, liquid nitrogen. Nitrogen is used. It is in the form of liquid, right? So please understand, liquid. Nitrogen is used in cryopreservation. It is in the form of liquid. So simply, liquid form of nitrogen is used in preservation of protoplast, cell organelle, enzyme, and extracellular matrix. Dear student, please understand, one of the agent is used in cryopreservation. Could you mention? Dear students, here we have discussed nitrogen liquid is used for preservation of following things: protoplast, cell organelle, enzyme, or extracellular matrix. Could you mention the chemicals used in preservation? Then what is protected? What are protected? Students are valuable. Okay, next we pass on to some cryopreservation protectants. Some protectants also used. Protectants. Some protectants are used, right? The one is disulfide, dimethyl sulfoxide. Dimethyl sulfoxide. One more, glycerol. These protectants, why are they protectants? Because these dimethyl sulfoxide and glycerol, which are used to prevent the effect of freezing, because when we use, just we are using minus 196 temperature, which is very extreme cooling temperature. When we protect the protoplast cells or tissue or any other organ or extracellular matrix in such a temperature, it is possible to be affected along with its viability, right? So in order to prevent such a effect from the cooling temperature, right? So simply these are dimethyl sulfoxide and glycerol. They are used to prevent, simply to prevent freezing effect, freezing from the cooling temperature, right? So that's why we are using some protectants, dimethyl sulfoxide and glycerol. So why are they used? They are used for retaining its viability and vigorous, right? In the nature of protoplast or cells or tissue or cell organelle. So this is the area to be expected in our examination. Mention the important protectants used in cryopreservation. That is the question. Please understand. Dear students, we have discussed some protectants which are used in cryopreservation. Could you mention, please? This is very important question. So let me ask you. Nobody is going to answer. Or let me ask.
Dimethyl sulfoxide, Glycerol or
Glycerol or sucrose.
Sucrose or glycerol. What are they actually generally?
Protectants.
Protectants. Could you mention the temperature used for cryo preservation?
Minus
190.
minus 196 degrees
Very
Celsius.
good. Just keep it up, right? Thank you for joining, and tomorrow we'll continue on the topic, right? Now I'm going to give you a home assignment. Please take down. Thank you.